From Microbe TV, this is TWiP, This Week in Parasitism, episode 197, recorded on August 13th, 2021. I'm Vincent Racaniello, and joining me today from Fort Lee, New Jersey, Dixon Despommier.
Happy Friday the 13th, Vincent. And it looks like a great day out there, but it's very, very hot. So I wouldn't call it a great day.
It's gone up a degree since we did TWiP. It's 34 Celsius.
Oh, you're kidding. Oh,
also joining us from New York, Daniel Griffin.
Hello. I like to think it's a little warmer because this is such a hot show.
There you go.
Indeed. And from Glasgow, Scotland, Christina Naula. Welcome back.
Hello. Thank you. It's lovely here, only about 20 degrees. So.
Oh.
Wow.
So, yeah.
That's pretty chilly.
It's good to be back. Yep. Well, it's just
It's comfortable, right?
Yes.
Yes.
It is, yeah.
Is it raining?
No, it's been a sunny day, windy and sunny.
Windy and sunny. You hear that, Daniel?
Windy and sunny, yeah. A bit
good.
of rain in between.
Perfect.
There's always a little bit of rain in Glasgow.
This is true.
Yeah.
Are you teaching this summer at all?
Not teaching now. I've been doing a lot of marking, but we'll be starting again soon. Beginning of September.
You going to have in-person classes?
No. So almost all my courses are online. We do have a practical week, which we hope to be able to run. I think we have distancing limitations, so we can't have a full class, so we might have to just split it into two.
I see.
But that will be in December. And our usual live teaching class that we tend to do in January, I think we've moved online now. So.
Wow.
So you guys is everyone masking there?
Here in Scotland, we do still have a mask mandate indoors. So in shops, secondary school children have to wear masks. In hospitality. So it's fairly good. But other than that, it's really quite normal.
It's a decent country. They have a mask mandate. Like here, nobody can agree on a mask mandate, right?
Yeah. It's a very small country, though, so we don't make much of a difference.
I don't think so. I think it's a good example. All right. Let's get on to our case from TWiP 196. Daniel, remind us what's happening here.
All right. Well, hello, everyone. We are going to be reminding those who, it's been a little while, and a brief update for those of you tuning in for the first time. On our last recording, at the tail end, we presented the mystery case of a 30-year-old woman who reported having areas of swelling in different parts of the body. Random different parts. She then got some blood work, and the blood work showed elevated eosinophil. So we probed a little bit more, and she shared that she had been traveling around Southeast Asia. When she got back, she had some loose stools, reported some tingling in the fingers, otherwise felt fine. Delved a little bit more into that experience in Southeast Asia, where she reported that she was an adventurous eater, trying all the local cuisine, lots of fish, pickled fish, salads. She does tell us that previous to this, past medical history, she was in good health. She had not had any surgeries, no significant family history. She did not take any medications. She lives alone. She is in the New York tri-state area where she lives. Where in Southeast Asia did she go? She went to Indonesia and Thailand. She is a non-smoker. She drinks alcohol occasionally, but does not sound like there's any sort of excess issue there. She is HIV negative. On exam, she actually did have a swollen area on the arm, and a biopsy was done. And I shared with everyone last time that the pathologist reported that the cross section had what appeared to be a larval nematode. And I left it there with people to tell us what they thought this might be, and then feel free to offer some treatments.
All right, Dixon, you want to take that first one?
Well, certainly. Anthony writes, the symptoms are a bit vague, but I feel like the geography matches up with Gnathostomiasis spinagerum. These worms seem to just wander about the human body as humans are a dead-end host. The CDC recommends if their patient is not that ill that they get ivermectin 200 micrograms per kilo orally in the morning or night for 2 consecutive mornings or nights. I have been starting my CPHT internship and have been filling prescriptions. So far, nothing parasitic, but quite a lot of thrush cases needing fluconazole. Hope Dixon comes back soon. Well, I'm back. Thank you, Anthony.
So Daniel, he's getting ivermectin. He wants to give ivermectin. That'll be protecting them against COVID, right?
We are still waiting for really good data to tell us whether or not ivermectin has a role. And it's been 20 months. And, as I think I've shared with our listeners, Active 6 hopefully is going to really address this because it is one of the most widely used medications. And it would be nice to actually really know if that is helping people.
Also.
Of course, I said that with tongue-in-cheek, listeners. Christina,
Yes.
Can you take the next one?
Alexander writes, Dear professors, first and foremost, I want to wish Professor Depomier a speedy recovery and good health. With any luck, he might now be reading this letter himself or have it read to him, fully restored and healthy again. On the case, the history of ingestions of incompletely cooked fish in Southeast Asia, the wandering edema, and the blood eosinophilia are all consistent with Gnathostomiasis. The larvae are acquired by ingesting raw fish, most commonly eel in Thailand, and the third stage larvae travel throughout the body, unable to complete their life cycle in humans by returning to the GI tract. Therefore, no eggs will be found on stool examination. As the diagnosis can be made by tissue staining in this case, serologic testing might be omitted in favor of early treatment. Albendazole, 400 milligrams twice a day for 21 days seems to be the standard, although ivermectin is an alternative. Thanks for the great case. I am anxious to hear the next one. Best regards, Alexander.
Daniel.
All right. Andrew writes, Kia ora from Pongaroa. No book one yet, but I would prefer to hear another one of Kevin's amazing emails, to be frank. My guess for the 30-year-old woman with swellings under the skin is Gnathostomiasis spinagerum. My PDF PD7 tells me that exposure is through infected fish, snakes, and birds. Our patient is an adventurous eater and has been in Southeast Asia where the parasite is commonly diagnosed. Humans are not a definitive host, so the larva will not mature, but they can cause damage, especially in the nervous system. After ingestion, the larvae migrate through the stomach wall, and apart from some tiredness and diarrhea, this stage is fairly asymptomatic. Once the larvae have migrated to the skin, subcutaneous swellings appear. Treatment, albendazole or ivermectin, hoping that this will be read out in the no BS level incubator. From Andrew.
So no BS.
I should point out the BS stands for biosafety, just in case.
I think this is a joke made on our live stream that we should call it the no BS incubator. It's great. It's a great joke. All right, Gabriel writes, hello from Baja, California. Cutaneous gnathostomiasis caused by Gnathosoma species is suspected by intermittent subcutaneous or cutaneous migratory swelling, elevated blood eosinophils, and relevant exposure. Confirmed by biopsy and microscopy. The loose stools and itching can be symptoms of the larva's intestinal and cutaneous phases. The tingling sensation on the fingers has been known to happen when inflammation during a roundworm infection causes medium nerve compression. Identification of the larva species can be performed by differentiating microscopic characteristics at an anatomical, cellular, or tissue level. DNA and other molecular tests can also distinguish amongst the 4 or 5 species known to infect humans. I'd bet on G. spinagerum, endemic in Thailand and also found in Indonesia. The patient was most likely infected by sampling local cuisine containing raw to undercooked infected meat from fish, frogs, snakes, birds, and other animals. A salad with a piece of infected snail could also do it. Thank you, Gabriel. Dixon.
You are muted, Dixon. The most common 3 words of the pandemic.
There you go. Okay. Okay, let's start again. Martha writes, Dear TWiP team, I hope this finds you all well. And yes, I am well. And I hope everybody else is too. Since I won a book already, don't enter me in the drawing. However, I intend to continue to respond to these cases. I am happy to report that I have no personal experience with this month's parasite. The patient presented with swellings on different parts of the body. History significant for travel to Southeast Asia and eating the local cuisine, including uncooked freshwater fish. On blood work, the eosinophils are elevated, and a biopsy from one of the lesions suggests an immature nematode. The CDC has a helpful page on Gnathostomus spinagerum, and she lists that. G. spinagerum seems to fit all the features mentioned. This is another case of humans not being a usual or functional part of this creature's life cycle. We humans get involved when we eat the second intermediate hosts, fish, eels, that's a fish, frogs, or one of the paratenic hosts, birds, snakes, or lizards. The larva is not able to complete its maturation in humans, so humans do not transmit this parasite. This is treatable by 3-week course of albendazole or a 1 to 2 day course of ivermectin. This is preventable by cooking fish to 140 degrees F, 63 C, which kills the parasite, or freezing to minus 31 F or minus 35 C. Best wishes, Martha. P.S. I learned a new term, paratenic host, an organism that harbors the sexually immature parasite, but is not necessary for the parasite's developmental cycle to progress. And that is correct. That is exactly what paratenic means.
Didn't we mention that on TWiP today, Dixon?
We did.
So an insect that picks up and mechanically transmits a virus would be a paratenic host, right?
That's right. Exactly right.
All right, Christina.
Cheryl writes, Vincent, Daniel, Dixon et al. Here we have a young woman with a history of travel in Southeast Asia who is an adventurous eater. She presents with arm swelling, loose stools initially resolved, eosinophilic. Biopsy shows larval nematode. My first thought was that maybe she tried the jumping salad in Thailand featuring delectable live raw crustaceans, minnows or crab eggs, i.e. nematode Capillaria philippensis. But wait a minute, symptoms for infection with said jumping salad a la nematode would have been vomiting and continuous protracted diarrhea. This is not our case. Next, I wonder, could this be loiasis, the infamous eyeworm? A possibility, perhaps. Filarial nematode inducing calabar swellings, that is, itchy swellings on the body. We have no report of itching in the subject case. But what about that swollen arm full of nematodians? Loiasis is vector-borne by deer fly bite. Our case hints suggest we need a foodborne parasite. Loiasis is found in Africa, wrong area of the planet. We need a Southeast Asia endemicity parasite. So we have to reject the loiasis nematode idea as well. Third guess, cutaneous gnathostomiasis. Eosinophilic? Check. Intermittent migratory swellings on torso or upper limbs? Check. Nematode? Check. Foodborne? Yes. Can get it from raw or undercooked meat, dishes with raw fish like sushi and ceviche, or even snake, duck, or frog. Endemic where? Japan, Thailand, Vietnam, Mexico, Southeast Asia. Bingo. This has got to be it. Very interesting emerging zoonosis. I'm including the CDC link, and she provides the CDC parasite A to Z Gnathostomiasis link here. Thank you so much, Vincent, Daniel, and Dixon. Others have thanked you far better than I, and I agree with all the kind, witty, smart, learned, erudite, and heartfelt appreciated expressed by so many. Thank you guys for being my science friends. Stick shift always a must for me. too, by the way, Vincent. Cheryl.
Lovely.
I always drive for shift.
Well,
stick
in Europe,
shift.
everybody does, right?
Yeah.
Here in the
Yep.
U.S., it's rare, but I've always driven a stick shift, and they're going to be taking it from my cold, dead hands one day. Never going to get it.
Yes, Charlton.
That's because all the cars will be electric.
Yeah, I know. I won't drive anymore. I'll walk. I'll take the
There you
train.
go. That's right. That's a stick shift train, I'm sure.
Daniel.
Yeah, I was remembering one of my first trips to England where they gave me a stick shift, but the sticks on the left hand. And
I've
I
done
thought
that
for a moment, and then I went back in, and that was all they had. So that became a very interesting, shifting with the left hand, driving on the side of the road that the Brits like to drive on. It was interesting. It's an interesting trip. All right, Lawrence writes. More of a Googling cap than a thinking cap. A search for nematode, parasite, fish, human, swelling, Thailand, Indonesia resulted in several candidates, but the best fit by far was Gnathostoma. It ticks all the boxes, including possible loose stools early on, elevated eosinophils, and no onward transmission due to not being able to complete its life cycle in humans. Treatable with albendazole, and if the COVID vaccine hesitant haven't already bought up all the stocks, ivermectin. All right, he gives us a link to the CDC site as well.
Nice. Excellent. All right. Katie Jane writes, hi all. In my completely non-medical opinion, we're dealing with two different things here. The loose stools are potentially caused by the woman's adventurous, fishy diet, but do not seem to be causing her much concern. I'm going to guess they're a red herring and unrelated to the eosinophilia and tingling sensations described. I'm not sure if the tingling in her fingers was bilateral, but I'm going to guess that it's caused by nerve compression of the nematode larva, which was found on biopsy. The nematode larva could be of the soil-borne species, but larvae of these species tend to invade more through the feet. I'm going to guess at a mosquito-borne Diphalaria species, which would be more likely on the arm, although presumably humans are aberrant hosts for these little critters. If this is the case, this woman's travels are not the problem, as this problem could have been picked up at home. All a complete guess, so I apologize for the rushed email. I really hope I get it in on time. And Katie Jane is from North Central Technical College in Wisconsin. Dixon. Dixon, you're muted again.
I try to do that so that you don't hear my heavy breathing. And then I forget to unmute. It's true. Skylar writes, good morning from the unpleasantly warm nation's capital, Washington, D.C. I admit I have been a major slacker and have not written in since spring. But with the third semester of medical school beginning, I'm hoping to jump back on the TWiP bandwagon and maybe win a book. Hopefully, I'm sending this in time to make it to the episode. We have a patient with changing swelling in her arm, known to be caused by some larval nematode. After returning from travels to Thailand and Indonesia, big hint is the episode seemed to be the importance of the patient eating raw or pickled freshwater fish. According to Leo and colleagues, 2020, gnathostomiasis caused by various Gnathostoma species is a foodborne illness seen in Thailand. It is caused by the consumption of raw freshwater fish or other animals contaminated with larvae and is characterized by migratory cutaneous swellings and eosinophilia. The paper is an interesting review of the disease and describes it as an emerging global disease as cases are rising due in part to adventurous eating. He lists an internet source. I'd like to give a more extensive differential, but I need to get this in before the next episode, and I need to prepare for the anatomy lab. Wishing you the best, Skylar.
Christina.
Sergio writes, hi all at TWiP. Case study 196, Gnathostomiasis infection. I think she was infected with Gnathostoma because she ate pickled fish. That's the type of food, animal, this parasite likes to reside. Once ingested, after 2 or 3 weeks, it travels through the stomach wall and reaches the skin, causing the swellings. I hope none reached her brain or eyes. Thank you for all your hard work. Love you all from Microbe TV. Sincerely, Sergio. P.S. I hope to visit the incubator one day once it is open.
Sure. You're welcome to do that. All right. Now, I just checked our email and we got one more. So, Daniel.
I see you added Elise. Elise, we're glad you snuck that in at the moment, right? Is this
got a big one?
That's
You got
what
a
we're
big
doing?
one.
Dear TWiP trifecta. I guess we now need to make it quadrifecta. So at least we'll have to update. But I very much hope this finds you all well. I'm writing to you from an extremely warm and humid lower Manhattan where it is 93 degrees F, 34 C. And my phone has been alerting me with warnings about excessive heat, air quality, and whatnot. Before I attempt a diagnosis, I wanted to tell Vincent about a local group that does whale watching, which is something he was talking about on a comparatively recent episode of TWiP. The Gotham whale folks do research and take people out on whale watching excursions. Their website is gothamwhale.org.
Cool.
So I am, as usual, unsure of my diagnosis because Dr. Griffin said this was a challenging diagnosis, and I am not super secure with my assessment. I suspect that the Adventurous Eater with the peculiar swellings and larval nematode may have a Gnathostomiasis infection. Gnathostomiasis is common in Indonesia and Thailand and also in the Americas and is easily acquired by eating raw or undercooked fish, though it is present in other protein sources as well. An infection is usually characterized by elevated eosinophils, though this is not necessary for a diagnosis. While the young woman's early symptoms, loose stools, tingling fingers, are symptoms of other infections, the random seeming swellings that appear, recede, and recur elsewhere are characteristic of a Gnathostomiasis infection. According to one source, undercooked Asian swamp eels are a particular source of Gnathostomiasis in Thailand and Vietnam. Depending on where the nematode takes up residence in the body, the infection can be more or less severe and dangerous and can cause encephalitis or meningitis. Since one of the swellings on the woman's body was biopsied, it should be easy to diagnose. The remedy for this infection is a course of albendazole or ivermectin, quite a long course, 21 days, and surgical removal of the larva. If this diagnosis is correct, how would the removal of the larva work for the patient since the swellings have come and gone? As always, thank you so much for everything all of you do. I'm so grateful for all of you for your work, your humor, your interests, and for sharing them. Many best wishes, Elise McAdam in Lover. It says Lover, Manhattan. I think that was one of those, Yeah. You're changing that to lower. That may have been, when I was tweeting during the boat race at one point, and I think it was like a slip of, I said, it's a dark and story night, just thinking instead of stormy, that it was also going to have the double meaning of lots of stories from the night. But I should share at this time. I know when I was listening to the TWiP discussion on whales, I don't know if people are aware, but it is whale research that drove my daughter Daisy away from science.
Really?
She was doing a project. They actually do a lot of whale research, the WCS, the Wildlife Conservation Society, which has people down at the aquarium in Coney Island. And she was going out on boats. They were doing that where they would dart the whales, where it bounces off and they get a small DNA sample. But her main job was to sit there with headphones on and listen to the acoustic buoys. Yeah. And then when she would hear a whale sing, she would have to mark it. So incredibly isolating work for a teenager to just sit there with headphones on by herself for hours, just listening to hear a whale song and tag it. And then once she had tagged the whale songs, then the senior people would focus just on the whale songs. But it is amazing, I have to say, to listen to whale sounds, songs.
Agreed.
I read that the sperm whale can make the loudest, can be almost heard around the world, right?
Yes. The hypothesis is that they can actually kill their prey or stun them with that sound.
That sound. Wow. Yeah, I'd like to go do some whale watching. That's.
I would too.
That would be fun. Okay, that does it. So it looks like people are slacking off for the summer. Huh? We had, I don't know, 10 or 11 guesses. Sometimes we have 20 or 30.
It was a stumper of a question.
Maybe it was so hard.
Yeah, that's exactly right. That's exactly right.
All right. So what do we have, Daniel?
Well, I think we still have 3 more people that didn't guess yet.
That's true. You are correct.
So shall we start with Christina?
Yeah, so I think I agree with all the writers. That was the first nematode that really came to my mind. I think what gave it away for me was the eosinophilia and the swellings, the kind of randomly appearing swellings. And, the adventurous diet was another pointer that made me think of Gnathostomiasis. So that would be my guess as well. I wouldn't go as far as settle on a species, but one of them.
All right. Now, Dixon, are you familiar with Gnathostomiasis? Do you concur?
I am.
What do you think?
And I do concur. In fact, I think there's probably very few other choices that you can make with this diagnosis, especially when you found the larva under the skin. And you could do a DNA staining with it because they have the genomes of a lot of these nematodes already done. So you could actually find out right on the section as to what species it is. But I think given all the other hints that you put into the case history, this has got to be Gnathostomiasis.
All right, Vincent, you going to go out on a limb?
Well, originally when I heard fish, I was thinking of anisakis, but having the worm in the hand isn't consistent with that because the anisakids go in your stomach, right? And they burrow into the wall, and you feel the pain and so forth.
That's true.
Right?
And besides, it's only from saltwater fish that anisakis actually comes.
Saltwater fish. I was wondering about that. So, I mean, these fish could have been.
These are freshwater fish.
How do we know? How do we know that?
We don't. That's true. We don't. But
it just
now
says
that we know what you caught.
It's just, yeah, of course, but it said pickled fish, so I didn't.
Yeah.
Right. No, you're right.
No.
You're right.
But the other thing, Dixon, isn't it the case that you wrote a story where a guy somewhere, someone put a poultice from a frog or a fish on their eye and they got Gnathostomiasis as a consequence?
No, it was a tapeworm. It was a larval tapeworm.
It was not Gnathostoma.
No, it was Spirometra.
That's right, Spirometra.
Yeah,
okay.
Spirometra mansinoides.
Okay, so they put the poultice on their eye to do, and then
the heat from your eye actually caused the worm to migrate out of the skin of the cold-blooded vertebrate into the warm-blooded vertebrate.
Wow. Okay. Anyway.
Yeah, there's, I think, a certain area of China that has that endemic.
China. Yeah, the Hunan Island has got like 30% of the native population has this infection.
All right, so I didn't guess Gnathostomas, so
but everybody else did, so I think you should follow suit.
Yeah, I would. I buy it. It makes sense. I mean, I like this guy who ticked off all the.
Yeah, that was terrific. That was a good one.
This was Cheryl, the attorney. It figures an attorney, you're going to tick everything off, right?
That's right. That's right.
Well, that was the impression of the pathologist, and this individual, to sort of close it out, was treated with albendazole. There's an ivermectin shortage, and for 21 days, twice a day. It's actually in the States. This is quite an expensive therapy. These uses are much less expensive outside the U.S. And the swelling and the eosinophilia all resolved, and the individual did quite well. So, nice, happy ending here.
Yeah.
All right. Well, before we go on, let's give away a book.
Okay. Would you like a drum roll?
I'm just wondering, Daniel, I guess we can bring the books down to the incubator.
That's right.
Yeah, I've got to swing by Columbia, so maybe I can swing by. We'll grab a whole bunch of books, we'll bring them down, and we'll get them all signed up.
Well, I've signed them already, I think. Is that true?
You have? Okay.
No, I think so.
I have.
Yeah, I've got to update my my ID card.
If not, I'll swing by too then.
You can see they're up there. on the
I
shelf.
see them.
So
Right.
here's one. Let's see if Dixon is telling the truth.
No, I said I think I signed one. Didn't sign one.
Let's see.
Didn't sign it. Boo hiss.
Maybe it's just an issue with the Zoom, but I'm not able to see anything.
I used invisible ink. I'm not sure that you knew that.
Not signed, Dixon. You're going to have to either.
No, I'd love to come in and do that.
Yeah, I can bring them to you, Dixon.
No, you don't have to do that.
I don't drive anymore. I forgot.
The mountain will come to Mohammed.
All right. Yeah. So anyway, the winner of this book. In fact, for the books who that have been won since the beginning of 2020, we owe it to you. Hey, hang on, Dixon, just a minute.
Oh, all right.
I don't even know how many. I got 9 people here.
You get the 10. You've got 10.
No, where do you get 10 from?
Wasn't that the last one?
At least won a book already.
Oh, all right.
All right, are you ready? From 1 to 9. How can it be 1? How could it be 1?
Okay, stop the drum. All
right, it's 1. It's number 1. It is Anthony.
Oh, that's
Anthony is the winner. So, Anthony, send us your address, TWiP@microbe.tv. If you are out of the U.S., then send us a phone as well. We need that for the post office. Okay. There you go. So let's do a paper.
Yeah.
And for today's enjoyment, Christina has picked a paper for us. So, Christina, tell us all about this.
So I have chosen, because we have been talking about vaccination a couple of times, malaria, and actually this paper came to my attention when I was listening to another podcast. I do listen to all the TWiP Microbe TV podcast, but I do also listen to others, and that came to my attention in the Nature podcast at the end of May. And I thought, that sounds really interesting, so I'm going to pick up that paper and have a closer look at it. So the title is An Invariant Trypanosoma vivax vaccine antigen induces protective immunity. And the first author, actually I think there are I never quite know. Never know who's the first author and the second.
I think it's just one on this one. It doesn't have.
I think it's just one. So I think it's Delphine Authorman. I'm probably not pronouncing that name correctly. And the corresponding author is Gavin Wright.
Unusual to see a single author.
They're based
paper.
in the Sanger Institute in. I should really know where that is, shouldn't I? Somewhere down south.
Hingston.
Yeah. Oh, yeah, there it is.
You don't know where that is?
I'm a bit patchy with my English geography. I actually thought the Sanger Institute was in Cambridge, but looks like I'm completely wrong.
Well, there might be multiple ones too, right?
Maybe, yeah. It could be, yeah. So that paper really caught my attention, firstly because we were discussing a malaria paper before, and then also I actually my PhD, I worked on trypanosomes, Trypanosoma brucei, so I kind of, when a paper in Nature pops up on trypanosomes, I'm very likely to go and have a look at it. And I actually thought, back then when I did my PhD, kind of a vaccine for trypanosome infections was something that we just couldn't really imagine it would ever happen. And that's really the reason why I chose that paper. So Trypanosoma vivax, I'm actually not extremely familiar with this particular trypanosome species, but it is a animal trypanosome species. It infects wild animals and livestock, and actually causes really big economic losses and issues in parts of the world where that trypanosome resides. So, and that is in sub-Saharan Africa, but you can also find the infection in South America. And what is quite interesting about this particular trypanosome is that, so the trypanosomes that infect humans, the Trypanosoma brucei, rhodesiense and gambiense, they are always exclusively really transmitted by tsetse flies. But this Trypanosoma vivax, so in the tsetse belt, it is also transmitted by tsetse flies. But in South America, where you do not have tsetse flies at all, it can be transmitted mechanically by other biting flies, really. So I think that's the paratenic host again, isn't it?
Mm-hmm. Yeah.
So I thought that was really quite interesting. So yes. So the reason why it is really difficult to develop vaccines against trypanosome infections is that they are essentially covered in a coat that is consisting of a single protein, the variable surface antigen. Is it a variable surface antigen?
Yeah,
I think so.
The
VSG,
the
glycoprotein, yes.
Glycoprotein.
Yeah. Right. And this actually is really immunogenic. So, the host would raise an immune response and would essentially get rid of all the parasites. But then every now and then, one of them would change the expression of that various surface glycoprotein coat, and another variant would then arise and multiply and cause another kind of wave of parasitemia. And so it's been thought really that other potential antigens on the surface of the trypanosome are maybe not so accessible, is really densely packed. But I think I read in that paper, actually, it's maybe more complicated than that. It's not really just, so it maybe also suppresses the immune response. But I'm not an immunologist, so I'm not going to burn my fingers here by trying to explain it. But if anyone else wants to jump in, that would be...
No, Christine, I think you're right on. I mean, this is one of those organisms that, is the paradigm of immunology. I think we've discussed this before. There's a cassette with different forms of VSG. And so there's dominant expression of one. And then once you get an immune response to that, it shifts over to another one. And we sort of talked about the mechanics of the shifting. And everyone always thought, well, that's really what the immune system needs to target. But it may be that the VSG has the ability to prevent us from targeting these other sites, which may actually be accessible. But it's that VSG that is thwarting our immune system. So this is there's a lot to this paper.
I think so, yeah. It's actually a massive, massive amount of work in that paper, and I think we're just going to scratch the surface. I noted down here for myself that the VSG distracts the immune system, and I think that kind of, is probably sufficient for this level of discussion. So anyway, what they kind of... what they thought is, actually they say here that the VSG may also subvert natural immunity by preventing the acquisition of high titer antibody responses to protective antigens, suggesting that eliciting prophylactic unnatural host immunity by vaccination could be achieved. So based on that, what they really did in the first instance, they really scanned the genome for potential candidates, and they had some selection criteria, one of them being that there shouldn't be any gene duplications, I think. And they also were looking for proteins that were more than 300 amino acids long extracellularly. And obviously, evidently, that they would be expressed in the mammalian stage of the life cycle. I've actually been thinking about the 300 amino acid size. I wasn't quite sure, why that? Is it because of the size? Is it kind of a similar size? But I haven't actually had the chance to look up the size of the extracellular part of the VSG.
Remember, they're looking for cell surface proteins, so they say it should be big enough to be accessible.
Should be big enough to really be accessible. Yeah, so they did identify 60 potential candidates, which they expressed, but they were able to actually get enough protein to work with from 39. And 34 of these 39 didn't really have an effect at all on the immune response. So they whittled that down. Actually, and what I wanted to say, the way they checked the parasitemia, I don't know where I've been living in the past few years, but they actually had bioluminescent mice to measure the level of parasitemia. And I've just never heard of that. That really
it's actually
my mind.
Yeah, it's actually the T vivax has a luciferase expression. So you can actually, what you're seeing when the mouse is glowing, it's the trypanosomes that are actually
so it's
glowing.
not actually the mouse that is glowing in the dark, but
he is
call them
glowing.
bioluminescent mice. But I thought that was really astonishing and actually seems to be really quite sensitive. So they did identify several potential candidates and did some further analysis of these and then settled on one variant that really, so a couple of the variants just delayed parasitemia and the mice would then develop the parasitemia and eventually die. And there was a couple that they looked at, and I think it was V23 in the figure 1. I don't think this is an open access paper, so it's maybe a bit unfair to refer to the figures. But so they were able to show that this antigen can elicit a strong immune response in the mice. And they were also, I think I'm just going to be quite brief with that because, again, there's a lot of immunology there. So what then they also did, first of all, they called that protein IFX, which stands for invariant flagellum antigen from T vivax. So I think the short name is quite accessible, I think. And so they did first some studies to kind of find out where that protein is localized to, and it localizes actually along the flagellum of the trypanosomes in the attachment zone. So the flagellum of the trypanosome is not kind of a loose flipping tail; it's kind of attached via the undulating membrane, and this is in that area of the parasite where that protein seems to be localized. Hence the complicated name. And yes. So they did some kind of analysis. They tried to figure out what would be the protection. How is the protection? How can that be explained? And so they did some complicated depletion experiments, and they did establish that the immune response was primarily antibody mediated. I think I'm getting that right, but not exclusively, as we'll see later. And what else have we got here? So they did also then do some passive immunization experiments. So they took serum of the mice that they had previously immunized with that IFX, and they were able to demonstrate that would also provide protective immunity. And I find that really astonishing because it's not trivial. They actually made lots of monoclonal antibodies. And they, did analyze a number of those, and they found some monoclonal antibodies that could also provide protection. And when they had a closer look at these antibodies, they actually realized they were all of one particular class, the IgG1 class. And that doesn't seem to be, I'm going to get into hot water now. That doesn't seem to be, this type of antibody doesn't really recruit other immune effector functions like complement. And I presume it maybe doesn't recruit T cells, but I don't think they've actually stated that. So I wouldn't really want to speculate on that. So what they then did, they thought, well, we're going to modify those antibodies, and we're going to turn them into IgG antibodies that have these recruitment functions intact. So they turned them into a different subclass, and they actually observed that these had a really substantially increased potency compared to the original monoclonal antibody that they looked at. So that demonstrated really that, that if the complement recruitment is actually a part of the mechanism of this protective process. I think that's it, isn't it?
I think that's a
Do I
good
get
wrap.
the immunology right there?
I think so.
I think so, yeah.
Well, the IgG2 will do better at binding FC receptors, right?
Yeah. Yeah.
So they think both complement and probably some other, because both changing the complement binding site and changing the FC receptor has an effect. So they think there's more than it's complement and probably something else that's going on.
Yeah, so it seems to be dominated by the complement recruitment. Yeah. So, so I think.
But what's interesting is that obviously they're not going to give passive monoclonals to goats and cows. No. And so I'm not sure they changed the FC portion, so I'm not sure the relevance of that to what, them making a vaccine. It's interesting, but
I think they were maybe just trying to elucidate what the mechanism, the immunological mechanism behind that was. And they did some work actually also with adjuvants. So they were trying to see, could we then go back to the original monoclonal antibody, and could we make that more effective? And they were trying different adjuvants, and actually there is quite a marked difference. So they did find a couple of adjuvants that really almost brought the IgG1 monoclonal antibody up to a protective level of that IgG2 that they had created. So I think they got a really big playing field now, because they've got a protein that elicits an immune response. They have got adjuvants that are really useful, or they know that can make a big difference, so they can play around with that. And actually that protein, I think, is really highly conserved across all Trypanosoma vivax species. Mm-hmm. So that probably provides a quite stable platform upon which to develop a vaccine.
Yeah, I was thinking, Christina, that maybe this was like reviewer 3 at the end. It was you're going to try to publish in Nature, so we're going to make you do some extra work, because it was really a nice story, right?
It's beautiful.
They screened all these things. They show that with the, you're gonna have to look at the number, because it doesn't roll off the tongue, but with V23, you basically have sterilizing immunity, right? Now that everyone's heard that term for the last 18 months. And then they go ahead and they say, well, what's it binding to? It's binding to this area in the flagellum. And then, and they get rid of all T cells. Who cares? They show that you can transfer the antibodies. You get this effect. It's IgG1, so we know that it doesn't have effector function. And at that point, I was like, this is, it's a wrap. Time to start testing the IFX, protein subunit vaccines, or maybe you'll make an mRNA or whatever you want to do. Because I don't really see that you're going to run around. I mean, when my cow got sick, I would have, rushed in with the monoclonals, but that's not really the scenario here, right? I mean, you have 3 million cattle dying a year in sub-Saharan Africa.
Did your cow die of Nagana, Daniel?
No, unfortunately, it was the hoof and mouth disease.
Oh, that's not good. Yeah.
Yeah. So
they did look. So they were kind of looking in cattle, and it doesn't really seem to be, cattle that were naturally infected. With T-vivax, they didn't really. They were not immune reactive, so they hadn't actually raised an immune response against that protein. Yeah. So I suppose there's still quite a lot of work to be done. So I think it's actually a proof of principle paper more than anything at all, because finally you can say, well, actually we can make a protective vaccine against those trypanosomes, those pesky things that keep changing their clothing.
Yeah.
And maybe it kind of shows that the VSG is maybe not completely shielding the trypanosomes from antibody binding. So I think that, as a proof of concept, I think that must have been so exciting when they got that. They did actually see there was, they did use a goat. So they immunized a goat. I don't know how many. They didn't mention a goat, but maybe they just had the one. I don't know. It must be really difficult having bigger mammals to work with. And actually that goat was not protected. So this seems to be at the moment limited to mice. And I don't know in what way mice are different from goat, other that they are bigger and have a shorter tail. I really don't know what their immune system looks like. But I think they would really have to take it to cattle. And I imagine, doing a vaccine trial on cattle.
Goats are like little cows. They're like little cows. I actually have a video of my son Barnaby. We not only did my kids grow up milking cows, but at one point we taught them how to milk goats. And we have little Barnaby, probably 6 years old, milking a goat.
Mm.
Right.
Yeah. So I don't know what the next step really would be. They don't really say where they're going to go next, but I think the next step would really be trial in cattle. But I can't even begin to imagine how you would organize a vaccine trial in cattle. I mean, that must be extremely expensive.
You start with getting their permission.
Yeah.
This is all true.
Yeah,
so
they make horse vaccines all the time, and they're big animals, and it can be done. Yeah, so
no, I think isn't there a brucellosis vaccine for cattle?
I think so.
And that was an issue. I actually had out in Fort Collins, Colorado, where people may know I've spent a bunch of time. Occasionally, the vets would be trying to inject that in the animals, and they would accidentally inject themselves. Right?
Ouch!
Animals.
Wow.
And they would call me, not because I was the right person to call, just because I think they're embarrassed to call anyone else. But.
So I'm going to Fort Collins next summer, Daniel.
Oh, fantastic!
You have
to let me know when you're going.
There's a bat meeting out there.
Really?
And they actually invited me last summer, and of course they canceled it. So they said, we're on for next summer, and they want me to go out and do a TWiP. So if you're out, if you decide to go, Daniel, we'll put you on the TWiP. How about that?
Yeah, let me know. I used to be chief of medicine there in Fort Collins. So,
well, you know all the good places to eat, probably, right?
Yes.
Nice. Well, Christina.
So just to summarize, really, I thought, more than anything, this is a paper that gives hope.
Yep. Give hope to the cows.
Not only that, because actually I didn't mention that when I was kind of stumbling through my paper presentation. Actually, that particular protein, the IFX, is not a protein that is present in other trypanosome strains. For example, Trypanosoma brucei. Now there's a method that you could use, an approach that you could take, and you could potentially identify suitable candidates also in trypanosome strains that cause infection in humans. And, that's another reason why I find that paper really hopeful, and it kind of gives me a lot of optimism. So...
Yeah, I feel like we, just like our immune system, we're too distracted by VSG. And this paper sort of points out you should be looking in other areas. You can't see the wood for trees or the trees for woods. I never know which way around that goes. Forest for the trees.
Forest for the trees.
Forest for the trees. That's right. So when I was in my laboratory days, I used to sit in on a lot of trypanosome laboratory summaries, because Lex Vanderplug was in charge of that lab, and they were also working on some of our projects, so there was some crossover listening. And one of the conversations was, why don't we concentrate our efforts on the flagellar pocket? Because that's where all the transport occurs for the organism, because it's covered, like you said, with a coat, except for that one little place. And so all the transport for the amino acids and the sugars and all the other things that this thing needs in order to carry out its life occurs through the in the flagellar pocket. So there must be other proteins in that pocket that if you interfered with them, you could block the transmission. And I never heard any more about it, but that was their think piece. I guess they didn't have the genome sequenced at that point, so they couldn't
but
get
now
there.
you have the genome sequencing, and they did
that's right.
nice electron microscopy to really say this is where that protein sits. So you can now focus. You could focus your search and you could translate it.
Yep.
Because I think T. congolense is also causing that cattle disease, Nagana.
Yes.
That you could maybe, identify a similar protein. So I think this is really
there are some that would take an interesting argument against trying to save the cattle of Africa, because most of them are imported cattle, and the imported cows are obviously taking up room that the native animals could occupy. So I've also heard that they should be raising eland for meat, because it's a very large antelope, and it's apparently very tasty, at least if you're a lion. They love them. So there's no reason why people shouldn't get used to them as well, but they could never domesticate them. But
nevertheless, I think a vaccine approach would be really good, because that parasite is also in the wildlife.
Yeah, it is.
So, you'd always, you'd forever have to treat. I can't really imagine that they would reintroduce native cattle, because they're just not as productive, aren't they?
No,
in their meat
right.
production and their milk production. So I think having a vaccination, and you wouldn't have to continuously treat those animals with those drugs that are really toxic.
Yep.
So I think that would be really amazing, really. So.
Yeah.
Thank you, Christina.
Indeed. Thank you.
Welcome.
Daniel, you had another case for us, or have you run out?
Oh, I do. So, all right. I hope everyone's ready for this one. This is a fresh off the press now that people are traveling a little bit again. This is a gentleman in his late 20s. And he calls me because he has now for 4 weeks, had watery diarrhea that is just not getting better. Now, he relates that he recently, feeling that it was safe to do so, took a trip with some friends down to Oaxaca, Mexico. And he stayed. It's really funny. Like, I don't know, maybe they hang out too much with me, but they, Dixon shaking his head about the Oaxaca spelling.
Exactly. Poor Vincent. He has to get it now.
I fixed it.
And
nice.
he's Dr. Griffin, I don't know how I could have gotten sick. I stayed at a really nice hotel. All the fruits and vegetables, the salads, everything. It was all very safe. It was all cleaned. But while there, he developed diarrhea, and the diarrhea has persisted now for 4 weeks. Now, interesting enough, he went with 3 other individuals. They all stayed at the same place. I don't know if the other individuals were quite as impressed with the safety of the fruits and vegetables, but it sounds like this gentleman was trying to be a healthy eater. He does not feel terrible. He just feels tired. He has the loose stools, does not have a significant fever, but it's been 4 weeks. He is not getting better. And he asked me for some insight. Past medical history is actually a young, fairly athletic gentleman, no prior surgeries. He relates that there's no significant family history. He doesn't take any medications on a regular basis. No toxic habits. He'll have an occasional beer. And
would that be a Corona, perhaps?
Yes. Which I've now taken to drinking myself, mainly for the comic effect, even though I don't actually like the way it tastes. But anyway, that's an aside. So this gentleman calls me and I recommend one test. I say, I am going to set you up for one test. And the next day I call him with the diagnosis and the recommendation for therapy. He follows my recommendation and he completely resolves.
Cool.
Daniel, the other people who went with him, they were fine?
They were absolutely fine. So just this.
So you're implying that the diarrhea that he had before he went down resurfaced while he was there. Is that
No. So he goes to Mexico and develops diarrhea while in Mexico. He's there for a couple weeks on this vacation.
Oh, I see. This is okay, fine. Okay.
And then the diarrhea now, when he calls me, it has been 4 weeks,
All right.
over 4 weeks, and the diarrhea is not getting better.
Okay.
Is he HIV negative?
He is HIV negative.
And does he have a partner?
No, he is single. Single, unmarried.
Was he the only one that ate fruit and vegetables and salads?
Getting the sense from him, it was really funny. That was almost what he started with. Like, I don't know how I could have gotten sick because all the fruits and vegetables.
You're right.
Like, so you ate a lot of fruits and vegetables.
Too.
I did, but
he also ate other things, I presume.
He ate other things as well, right?
Like,
so we're in Oaxaca, so chicken, beef. He's not a strict vegetarian.
But he always ate at the hotel, right?
Yeah, he made a point of saying that he ate almost exclusively at the hotel, so he couldn't imagine how he got sick.
Almost exclusively.
You just feel safer, don't you? But that's maybe false security.
But maybe one. You said almost exclusively, so maybe once he didn't eat at the hotel, but
he may have had some other. Yeah,
but he didn't note anything different. Okay, very good.
So was the diarrhea just watery?
Really, just watery.
Yeah. Okay.
Not bloody, right?
No,
not bloody. No blood work at all on this?
No, we just went right to the chase. I ordered one test, and then the next day I called him. He was shocked that I had the results so quickly, and treated him. And a few days later, he started to feel better, and symptoms completely resolved with treatment.
Daniel, you can tell him you wrote the book on the subject.
Mm-hmm. Indeed. All right, that's TWiP 197. Pretty soon we're going to have 200.
Pretty soon.
Folks.
You betcha.
And I guess we'll just let it pass, right?
Oh, no. We're all going to go to Scotland and celebrate with tasting a fleet of single malt scotches. That's what I suggest.
Well, you could, single malt scotch. That'd be great. Absolutely.
And haggis. If we time it right, we can show up for
We could do the haggis. You
guys can have it.
You just don't have to address it.
Yeah.
That's true.
With Robert Burns, if you just eat it, not in January, you can just eat it and enjoy it without
I guess.
Try and pronounce the poem.
You should probably not tell us what's in it either.
Well, I actually eat a vegetarian one.
There's a vegetarian haggis?
Yes, there's a vegetarian haggis for people like me.
It sounds oxymoronic, but okay.
Indeed.
I mean, haggis is mostly oats with some animal, insides and lots of spices. So, yeah.
The single malt scotch will take all of that away from you, Vincent. So you'll lose your inhibitions.
You can have the haggis. I'll have a single malt in its place. How's that?
There you go. All
right. Show notes, microbe.tv/TWiP. Send us your case guesses. TWiP@microbe.tv. Let's go. More than 10. Come on, folks.
Exactly.
This is an easy one. You should be able to get it.
Indeed.
If you like what we do and you want to support our work, including the establishment of a new recording studio, please go over to microbe.tv/contribute and part with some of your hard-earned funds. It will be used very well. Dixon Despommier is at trichinella.org, thelivingriver.com. Thanks, Dixon.
You're welcome, Vincent. This was a lot of fun. I'm sorry that I didn't have a hero for this presentation, but I will next time.
Why didn't you?
The same reason that
we only got 10 guesses. Okay.
That's right.
No worries.
That's right.
Daniel Griffin, Columbia University Irving Medical Center, parasiteswithoutborders.com. Thanks, Daniel.
Oh, thank you. And everyone, go to Parasites Without Borders so we can help support floating doctors during this and the next 3 months.
Exactly.
Christina Noula is at the University of Glasgow. Thanks, Christina.
Thank you very much. That was really good fun. I hope I didn't butcher that paper too much.
Not at all. Not at all. What are you worried about?
It was fine.
When we talk about immunology on TWiP, we all put on our asbestos gloves so that we don't burn
I've
our
actually learned
fingers.
a lot of immunology on TWiP, so
Very good.
I think it's been really good because
the
I
only
did
one that doesn't do
immunology
that
at
is Brianne.
university, and I never really understood it. But now I think I'm slowly easing
Starting
my
to
way
make
into it.
sense. It's starting to make sense.
Starting to make sense.
Great.
Yeah, it was great fun. Thank you.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiP and Ronald Jenkins for the music. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.